Okay, so I'm Tyler, and I'm going to take you through the first part of our topic, which is how CRISPR is helping to shape the research and treatment of colon cancer. So I'll just start with a brief rundown of CRISPR. So CRISPR, or clustered regular interspaced short palindromic repeats, and the associated Cas proteins, are RNA-guided site-specific nucleases, essential in bacteria endogenously as an adaptive immune system. The Cas protein mostly used in labs is the Cas9 protein, which is taken from the Streptococcus pyogenes. The artificially constructed Cas protein has an associated CRISPR RNA, which has a 20 base pair sequence on it, which will bind complementary to a DNA strand in the host DNA. And it's very specific. It also has to be adjacent to a 5' 3 to 5 base pair protospacer adjacent motif. So there's another type of RNA called transactivating RNA, which binds to the CRISPR RNA in a hairpin loop, and that activates the entire complex and allows it to anneal to the DNA. And this entire RNA complex is termed the single guide RNA. So once the Cas9 protein has annealed successfully to the DNA, it'll initiate a double-stranded break or a single-stranded break, depending on the protein. And this will allow a artificially created repair template, to be inserted into the host DNA at a certain site. If there's no template available, the Cas9 protein will cut at the same site repeatedly until a mutation occurs at the double-strand breakpoint, and it can no longer recognize the sequence and cut it. So, anyway, colorectal cancer. So it's just simply a development of cancer from the colon or rectum. Some risk factors include smoking, lack of physical exercise, some diseases such as inflammatory bowel diseases, traditional treatments for colorectal cancer include surgery, radiation therapy, chemotherapy, and globally, colorectal cancer is the third most common type of cancer, and it makes up about 10% of all cancer cases. And it is more common in developed countries, where about 65% of all cases are found. Okay, so what I'm going to be talking about is how CRISPR helps research in terms of modeling conditions for the generation of tumors and modulating tumor growth conditions. So this study was it looked at phosphokinase C in mice. A population genomics studies revealed that most human phosphokinase C mutations cause a loss of function, especially in cases of colorectal cancer. So a correction of the loss of function mutation by CRISPR was found to suppress tumor growth in xenograft models, which basically are human cancer cells transplanted into a foreign species, which in this case was mice. Another example, a more interesting example, is looking at non-coding RNA. So in some cases of colorectal cancer, it's found that a certain type of non-coding RNA called long non-coding RNA, which is generally over 200 base pairs, is downregulated. So certain studies wanted to look at a mechanism to knock out the non-coding RNA. And CRISPR generally knocks out by a frame shift mutation, but that's not very handy with non-coding RNA because it doesn't have an open reading frame. So instead they used a dual guide RNA vector, which cuts at 2 sites on the DNA simultaneously and allows large sections of the non-coding RNA to be removed, and that knocks it out. There are other cases of modeling, such as inducing certain alleles that induce tumors, but my partner's going to talk about that later, so I'll let him know.
All right, so when we come to tumorigenesis, it's important to recognize that cancer doesn't just proceed if you have a single mutation in a cell. It requires multiple mutations or multiple hits, and that allows colon epithelium to transform into cancer. So according to the classical model, you start out with normal colon epithelium, and then you have generally a mutation in the APC gene, which causes an adenoma. Adenomas are benign tumors, so they're pre-malignant masses, and they don't necessarily turn into malignancies. But if you have following mutations in other genes, including CRAS and, for example, RAS here, you can proceed from an adenoma into a carcinoma, which can then, which is a malignant mass, which then can metastasize to other parts in the body. So it is actually quite a very complicated set of steps to get to an actual cancer mass. And you can see some of the pathways here. So mutations in any one of these genes can lead to a malignancy. Primary among them is APC, but WNT and this entire pathway is actually a gene that's quite familiar to us. It's actually the wingless gene in Drosophila. And it sort of makes sense that this would be involved in cancer in humans, and that's because in Drosophila, the formation of wings are related to embryogenesis. So if you have a mutation in a gene involved in embryogenesis, which of course is going to determine cell proliferation and cell migration, you might run into problems down the track in adult cells. And as you can see here, there's a multiplicity of different genes that can be mutated to lead to cancer. So in any one of these, and in fact you need a collection of more than one to actually get to a cancer. But you'll see here, for example, in 81% of cancers that are non-hypermutated, there's a mutation in the APC. So what we want to do is to model these pathways of cancer formation. And the way we can do that is by actually inducing these inducing these changes within cells to see if we can get them to progress through cancers. So Matano et al. Used CRISPR-Cas to generate mutations in intestinal stem cells. And what they've done is they've used 5 separate genes, APC, CRAS, MAD4, P53, and PI3KCA, and what they've done is they've mutated them separately, and then they mutated them combinatorially. So what you can see from the results is, over here you've got A, K. These just indicate that they've got more than one mutation. And they found the more mutations you add to that stem cell, the more able it is to survive under unfavorable conditions. So these factors at the top are actually inhibitors of growth. And you can see that they, in the case of the last cell line, they're growing regardless of all these separate inhibitors. So that's really interesting. But when they transplanted these cells into genetically engineered mice to see if they could actually metastasize and produce malignancies, they found that the cells were actually stable. So even though that they had features of adenocarcinoma, they weren't actually doing anything. They produced microlesions that didn't actually seem to get any bigger or progress. And they didn't continue to grow after 2 months, whereas, as we all know, cancer continues to grow. So the explanation for this really lies in the dark side of the cancer genome. So it's very easy for us to categorize small changes in genes. So single base changes, indel mutations. And although you need 5 to 8 of those type of mutations to get the development of the cancer, there are certain other mutations that certainly must be at play. And this kind of genomic dark matter includes rearrangements, copy number alterations, methylation, and what we can do with CRISPR is we can actually induce those kind of changes that we haven't been able to induce before using traditional means. So the way we do this is we can use 2 separate guide RNAs in the Cas machinery, and we can actually get it to snip the chromosome in 2 parts, and then it'll rearrange. And so you can have inversion mutations and you can have rearrangements. And you can also use enzymatically dead Cas to get the nuclease to not actually snip, but just stay in that place that you're targeting, and then you can get effectors to methylate the DNA at that site. So you can test these epigenetic changes. This pathway is really important. Elucidating these pathways are really important because it allows us to determine the specific mutations within personal cancers that we can use to find particular drug targets that will work for individuals. We already do this in CRAS, for example. We test CRAS mutations in solid tumors to find out if they've got mutations that will render them immune to the growth inhibitor cetuximab. But we can then engineer this and open up a new door in genetically, in personalized medicine, I mean. So in summary, this has huge potential, and it's been very difficult to model these pathways through traditional means, but we're getting there with CRISPR-Cas. Thank you.